All right. So my starting point for this one is a concept that we use, and I'm going to get a little bit more detailed about this as we go. But the idea is that all animals out there, all plants, etc., all macroorganisms are associated with microbial communities, viral communities, fungal communities, etc. And these are ecological units that compete for space and against each other in the environment. And the coral holobiont is my favorite system to work on. So the coral animal, if you're not familiar, is a cnidarian. So it reaches out and it has stinging cells that kills things like a jellyfish. It's just a jellyfish stuck in a carbonate skeleton. And then inside the coral animal are these algae, the zooxanthellae. And so these guys give most of the food, actually, that allows them to build the skeleton comes from these guys doing photosynthesis. And then living on the surfaces are a whole bunch of protists. And then right above the mucosal layers, corals are just all mucus, effectively, are these microbial and viral communities that are fairly specific. And I'm going to address this a little bit. And then inside the skeleton, there's another community, which is algal and fungi. And they'll live below the animals in there. The working hypothesis, or the working just model for a lot of things we do, we mostly work in corals and algae in humans. And the idea is that the holobionts are assemblages that really form ecological units, that they're readily adaptable to changing environmental conditions and so forth by switching membership or through horizontal gene transfer. And they often are competing against each other. And one thing that I'm going to talk about a little bit here is that they may be units of selection, mostly because of Paul's talk yesterday. And I figured I'd at least broach it, even though this is a...
Excuse me, so how do you define the boundaries of the holobiont?
Yeah, so on a coral, it's actually really hard because they fragment and you have different individuals. So usually just visually as an ecologist, when I'm out there, I say that's a holobiont. With us, it's easy because you're one and you're walking around differently than him. So that's exactly how I look at it. The problem is in the world of corals and a lot of these colonial organisms.
Even we aren't. Right? We're all connected and blah, blah, blah.
Exactly.
Is this clustering well defined?
No, it hasn't been. In fact, it really didn't start out as, it started out as just a construct to organize amongst this really unclear place and a word to use. And we stole the word from Lynn Margulis because she had been thinking about how symbioses were so important. She's talking about endosymbioses, and we just said, well, it's just everything that's associated with whatever we're going to call our ecological unit. Okay? Yeah, so there's no strong definition of biological.
You can't define it by boundaries, dominant energy flux between the components or gene transfer or anything like that?
Nothing like that. In fact, I'll go into, that's actually part of the confusion, or has been over the years. Right. And the place I'm going to work, most of my data is going to come from the Central Pacific, and these are the Line Islands. We live there. That's Australia down there. So these are about as far as you can get from humans. And that becomes important later on because these give you really pristine coral reefs to look at. This is a Kingman reef, and I want you to get, the things you should notice about these reefs is that coral is the main thing on the bottom. And then there will be these algae called coral encrustose algae, which are pink, and you'll see those in a little bit. And there's very few small fish when you look around. So this is starbuck. For the most part, you see big fish. So you see sharks and groupers and so forth. And all the small fish are hidden in the coral. So the structure of the reef is what gives the place for the fish to live. This is the highest biomass ever recorded for a marine system. This is Malden, which is one of the biggest, close to 100% coral reefs that has ever been described. This is Millennium, which is the biggest working atoll on the planet, and it's a very beautiful system, actually one of the most beautiful places I've ever seen. And this is Jarvis, and Jarvis becomes important as we go through because Jarvis sits just south of the equator. And again, it's essentially like only coral and this coral encrustose algae dominated by sharks and just a few small fish that you can actually see. And then just north of the equator in the same biogeography range is Christmas Island. And the difference between, and you can see that this is completely different, right? So it's only macroalgae for the most part on the bottom. And then these are dead corals for the most part that you see scattered out through here. And the difference, of course, is people. So you add people to coral reefs and they die. So this is what the thing goes. Christmas here, Jarvis here. And then northerns and southern line islands. You've got the biggest population here, second biggest on these two islands here. There is a marine station on Palmyra, and then Kingman is uninhabited, as are all of these down here. They're all uninhabited. This is what the nutrient regimes look like, and this you don't have to remember, other than just know if you're not used to oceanography, where you have the equator, you have what's called the equatorial upwelling, so nutrients are pulled up on this, and you get less nutrients as you go away from the equator up to a certain point. Okay. And these are the nitrogen species and the phosphate species is what we've measured here. All right, so a typical way that we would go about doing this is a metagenomic approach. And so what you go do is you go down and you suck a whole bunch of water off the bottom, and you're below the boundary layer, so you try to keep right on the surfaces of the benthos, collect them in these little cubits, we call them, and then we'll concentrate them to get microbial and viral DNA, and then we'll use that and sequence a whole bunch of it. And what I'm going to show you today is from 11 different islands where we've done multiple reefs on most of the islands, and it's about, I forget, this is usually about half of this, so maybe say 1.6 million reads that we're using to do the classification. This many got hits, and then we're going to divide them into predicted taxa, so what bacteria did they come from, and then predicted metabolic potential, so what genes are they encoding, what types of genes are they encoding. Is that good?
So
yes.
Going back quickly, the comment about the 800,000 or 1.6 million, so
yeah.
Half of the stuff you find, you don't know what it is.
Exactly, roughly, and I don't really remember exactly for this data set, but something like that. With the viruses, it'll be a lot less, it'll only be 10%, and I don't remember exactly how we did this classification, because it depends on, as you know, like the e-value we choose or whatever we are doing. We're using just the C defaults, I think, in this case. So what you find is this with the microbial taxa. So the types of bacteria that are living there are dependent on the bottom type. So that seems fairly straightforward, but it gets tricky as we go through and look at other things. So remember, Christmas is the one where it's just macroalgae everywhere. So the types of bacteria that we fit, this is just one of these MDS sort of plots. Christmas sits out here way by itself. And then these are the next two islands with the most people on them, right? And they're not as degraded, but they're shifted in the types of microbes that you have living on them. And then the uninhabited ones tend to form this other group, right? And it doesn't really matter what their biogeography range was, okay? So that's an important point one. And the drivers that amongst the things that we've measured are basically hard coral cover, macroalgae, which is big algae, and then turf algae, which is kind of the slime layer, if you're not used to it, that you see on lots of rocks in a coral reef or even in nearshore environment.
Excuse me, so it's
yes.
more diverse the further away it is from humans?
No.
Kind of one you should
No,
keep this slide?
it's not more diverse. No, actually, diversity usually goes up with people up to a point because of the intermediate disturbance sort of stuff. And that seems to be basically true with the microbes and the viruses too, roughly. Mm-hmm. And And the main players, you'll see it as we get, but remember Bacteroidetes is shifted towards the turf algae, and you get cyanos and stuff pushing this direction, and then Pseudomonases and so forth, and Interbacters and so forth are towards the macroalgae. All right. This is where it gets weird and where I think it's most exciting. So if you, instead of take and you look at what taxa do I have, you just ask what metabolism are they coding, it becomes independent of the number of people and the bottom type, and it becomes dependent on the biogeography, okay? In particular the nutrients, but the distances from the equator, etc. So you can see this. So here, remember Jarvis and Christmas are completely, I mean, this is the one that's totally destroyed, and this is the one that is really nice, and they're in the same they're the same nutrient regime, same sunlight, etc. Then these two islands are really close to each other, and etc. And so that these two are right next to each other out there. So what matters in the case of what they're encoding is dependent on their distance from the equator. You could think of it that way. Phosphate, distance from the equator are the strong drivers here. And this just gives you an idea of, if you break it down a little bit more and you just say, well, here are these different islands, and this is their distance from the equator, and what nutrients do I have that are associated with it, and these are just different broad categories, you get these correlations. So you get positive and negative correlations depending on where they are. So the working hypothesis, and where we are right now, is that the microbial course, so this is your ribosomal, the things that make you a cell, are really just selected by the energy that is coming from whatever the benthos is. So if you've got primarily a macroalgae, that's feeding a certain set of microbes, and that's what you culture up. And that the microbes that are in these conditions are adapted to basically an island by island sort of regime via a horizontal gene transfer events, right? Things like nutrient acquisition. Which means that what we need to really know is what's going on with the phage and the plasmids, which is the thing that we're doing right now. And we're trying to see are the genes that are positively correlated showing up in one of these two groups.
Excuse me.
Yes, ma'am.
Do you have any metatranscriptomic data on these islands to see whether there's a correspondence between the presence of the genes, of the metabolic genes, and their actual activity?
Right. So we have, a lot of chemical background in all of these systems. Anything specific that you're thinking of?
I was thinking if you compare the metatranscriptome with
Right, so what are they upregulating and that sort of thing. So we do have transcriptomes from some of this, but our main data sets that would be on the chemical side would be what the water chemistry is, and then what the metabolomes of the surfaces that we're sampling. Those are the two main things that we're working with. Okay. And we're still, honestly, those are hard to work through. So we are working through them. And I can show you that, but it'll take a while. So the idea here, and one of the main things that I want to determine from these data sets is, are the genes that we're seeing that are for local adaptation being moved through what I call the professional mobile elements? So your prophage, your plasmids, and your islands, right? Are they being moved from the phage and plasmid metagenome into the microbe metagenome? Or is it more these accidental ones where you get like a specific gene will get into a system and get selected up? And right now, it's leaning towards this side. So I think it's going to turn out to be the phage and the protists. The plasmids will be the most important ones for causing this evolution that we're seeing, or this adaptation that we're seeing. The other thing that I wanted to use this data set to look at a little bit is just to pose to people like Paul and that are interested in this. There's this big question out there: is a holobiont a unit of selection? And it's a group level selection that's been really hard to get at in any system, really. And as you probably know, well, as I know you all know, you need reproduction, variation, and heritability for Darwinian sort of selection. And this is the data from, this is the studies that have looked at the holobiont. Is it heritable, right? And the answer is that depending on who you are and what you want to see, you'll answer that it is heritable or it's not heritable, right? And this is in all holobionts. Doesn't matter. If you're in the human microbiome, you'll answer it one way or the other. There's not great statistical or even theoretical ways of thinking about this.
How is heritability even determined? Just asking about similarity.
Yeah, did I see the same stuff on the same organism over time or space? Is really what I'm saying in that case.
Without
Yeah.
regard to the rank abundance of those particular OTUs.
Right. So that's where we've gone, and that's where I'm hoping that we can make a good, or something that will at least get us to, can we even ask this question in a useful manner? So the answer is whatever you want it to be, flip a coin, right, would be, and it really is, it's almost exactly 50% if you count up the studies that have been done. And the thing that we've built is something we're calling the ubiquity abundance plot. And it really is what you're talking about. It's looking at the rank abundance and watching how they're moving and whether they're always found there, or would they be there if we sampled deeper, is the logic. So it's a statistical test. This is the sort of data you get out of it, and I'm not going to go through it because it's a little bit past me, but for you, the math nerds in the group, we've got this as the supplement that goes with it, and it'll explain it. And this guy is he's a very talented mathematician. So I guarantee the math is right. I don't know if the test is right, but the math. And it's what Nigel was alluding to, is that you're looking at how do things move in rank abundance curves from sample to sample, right? Which we've done a lot of over the years. And what you find from this test is that there's basically two main groups. The things that are sporadically associated with the coral holobiont, and things that are stably or obligately. Okay. The difference between an obligate is that's something that we find on 100% of the same sample, right? And they always tend to be in high abundances, okay, with very low diversity. To give you a feel, if you take the same coral species, you'll find one or two microbes that are always associated in what we would call obligate. However, using this test, if we look at what about those lower ranking things, and are they staying there, it turns out that there's a large community that's got high diversity, but low abundance, that is stably associated with those corals, right? And then we can show that. And then the sporadic ones are the things that seem to be falling into the system and then falling back out of it really quickly, right? So these are literally things just sticking to the coral. Okay. That would be the same if it was your gut or so forth. So one of my questions that I really would pose to you is that if it looks like this is heritable in the sense that we really do think that these things have these communities that are stable and obligate, could it be that this, the holobiont, minus the sporadic guys, throw them out, is a unit of natural selection? Right. Any questions about that part? I'm going to jump into some ecology.
What about the reproduction part then? How do they reproduce?
The coral itself, the holobiont itself. So that's a really great question. So there is some evidence, they fragment. So one of the main ways that they get around is to break into pieces. So you could imagine them moving their microbiomes that way. But they, of course, also have very detailed sexual versions of that. And when they do that, it depends on the type of coral. So corals come in two flavors with their sex. So a lot of them are spawners, and most of them are spawners, and they set up all their gametes. They meet in the water column at the top, and then they set up an organism, they find a place to sit, and then they expand out, and then they acquire their symbionts. In the other case, they're brooders, and they actually do internal fertilization, and then they crawl away from their parent. And when they do that, of course, they take both their zooxanthellae and their microbiomes with them.
So if this happens, to what extent would it be, some people draw an interesting distinction between growth and reproduction.
Right. Yeah.
And this sort of matters because if it's growth, evolution sees more of the same.
Right. Just continuous, yeah.
Just a
Yeah.
spread of grey squirrels throughout England replacing the red squirrels.
Right. Yeah.
Yes, so there's a group of grey squirrels, but really, I don't think that selection is working on the group of grey squirrels. It's sort of an ecological
Shift.
replacement. It's growth of grey squirrels. What's your sense here? So I can,
Yeah.
would one,
Yeah. My sense is that it is not a unit of selection, quite honestly. That's my gut feeling, but
that's some semblance of
Yeah.
possibility. Yeah, I think that it is these, I'm not sure, in this area, Peter Godfrey Smith has written a very clear book on Darwinian populations, where what he's done is emphasize the marginal nature, the fact that things are not black and white. They are sort of big organisms that we see, but once you go down to things as ephemeral and as loosely defined as coral reefs and so on, it gets fuzzy, it gets murky. It doesn't mean to say that selection couldn't work to some extent on these things.
right.
I'm totally with you, wondering.
So I think it's a very interesting question whether you really need reproduction for evolution. And I think you can imagine that you wouldn't necessarily need it in something like the hollow bones. I agree with what Paul was saying, but you're making the assumption that the thing is sort of spatially homogeneous. And if the holobiont is spatially heterogeneous, then it can have its own internal dynamics, its spatial structure, and those changes without reproduction can affect the population genetics, and so the thing as a whole can certainly evolve in the sense of what we normally mean by evolution without there being reproduction, because you've got spatial variability within the holobiont. So
I mean, I kind of agree with that. And something that Sylvia and I worked on last year. Actually, me starting off thinking reproduction is the key thing, and then coming down through thinking to recognize that actually you could have the minimum requirement is some sort of genealogy,
yes,
which may just mean you have sort of 2% recurrent each time. Everything else is mixed and altered. And so you feel, on one hand, it's not comfortable to call it reproduction. Reproduction.
right.
But there is a genealogy established.
I think it's not reproduction and growth are the sort of two end members. I think you also have dynamics as well. I mean, there's all sorts of dynamics that can happen for the phenotype and the genotype and their interplay, and you can imagine something evolving in that way without necessarily
Having a
I'm interested in those sorts of questions because of origin of life
yeah.
issues.
I think another aspect here is how these organisms obtain their bacteria, for instance. If you look at the zooxanthellae, we know that some zooxanthellae are tightly related to some specific corals, but very often corals can also exchange their zooxanthellae by taking them up from the environment.
Right.
And then, of course, you get a whole different dynamics, because there's more or less mixes of the zooxanthellae among the different
Right.
coral species.
And that's what's happening here. Remember, we know that that's happening. We're just saying that there is a group of things that are moving together as far as we can tell. But it would be similar to the zooxanthellae idea that just because we have bacteria A doesn't mean that bacteria A doesn't have different genes, even in the obligate symbionts, which I think you could easily argue are going to be under regular natural selection sort of behavior. Hold on just a second, Andy, yes.
I was wondering about just the definition of holobiont. So how do you spatially actually define it, because of course there's the entity of the coral, and
Yeah.
you can have this spatial diversity. But then actually what you are considering here is more like from island to island. And there you really have entities that are separated and there can be some
Yeah.
dispersal. So it seems to me that with respect to normal, groups that we think of as being subjected to selection, you have a bit of a problem in defining exactly what your entity, what your higher level would be.
So I agree totally with you, and I would point out to everybody is that these are basically the oldest animals that we have left on the planet. They probably are chimeric at the level of even their own DNA, right? So they're probably picking up things. We know they can form viable hybrids both sexually and when they're in their egg form, when they're in their blastulus form, they can fragment and then be put back together. So this is actually real evolution, but not matching anything that people spend a ton of time. They're not clonal, they do really weird things. So it does challenge the history, or the natural history challenges our normal thinking about how evolution would work. So yeah, I'll leave it at that. But it does, I hope it brings up some points of how these are really fun, especially, if you're interested in origin of life and origin of multicellularity, these are places to really think about. Andy?
Yeah, I just think out there on those islands that the fragmentation is way more significant than the sexual reproduction, right? If you don't have any advantage from the sexual reproduction in one generation, I think fragmentation totally outweighs any kind of sexual reproduction and those recombination. So you propagate the holobiont easily because it's just happening so much more often.
Through fragmentation, yeah. And the reason is that these things are so far apart and there's so much distance out there, it's hard for one coral to get to another island, right? So there's going to be coral population bottlenecks or isolation. All right, so that's my step, and now I'm going to jump in so that the ecologists don't feel unloved. So here is our system and our model of how these systems work. The coral reef itself, and remember, these are really spectacular structures. I can't emphasize this enough. This is like 5,000, 6,000 feet of coral headed down towards the ocean floor. This is what Darwin was talking about when he wrote about atolls. So these things are out in the middle of nowhere. It's only coral and coral encrustose algae. You can think of the coral as forming basically the rebar of the system. So it provides structural sideways shearing force. And then this is the cement, the coral encrusted algae. And you need the two of them to form these, which are the largest structures on the planet, biological structures on the planet. And this is what it should look like. It should basically be pink with some brown. There's also other players, which are these macroalgae, and they're very not apparent on a healthy coral reef. They're there, and you'll find a couple of them, but they're really spotty. And then these turf algae, on a healthy reef, will just look like, mostly like bare coral skeleton, right? Because they're chewed down so much. And the guys that do the chewing are mostly, at least in our system, fish. There's of course sea urchins also, but fish and little invertebrates come down and they chomp on the turf algae for the most part. And some of them suck the slime layers off the CCA. And then these guys almost immediately get eaten by these guys, which then get eaten by these guys. So there's a very fast movement of anything that's photosynthesized, either to build structure or to produce sharks. And what that means is that there's almost no energy left over that goes from these animals and the algae to feed dissolved organic carbon to the microbes. Right? So these guys are held in check. That's my working hypothesis. I think it's about right. Is that these guys are only being fed specialized things, and that's one of the ways that you're controlling them on your surface. And then the things that people do, of course, is we come and we fuck it all up by killing all of these guys. So you lose all of the higher trophic levels and you lose your ability to eat all of this food. Because remember, as I move through each trophic level here, I'm going to burn most of it off as heat. Then you end up with a system where these start to grow up, and then they feed more food to, more DOC to the microbes, which the microbes come back, they start killing off corals, which gives you more space for more algae, which gives you more microbes, and you get in what we call the dam loop. So this is a positive feedback system that destroys a reef. And it seems to happen really quickly. There are examples we know of where a coral reef will go from apparently healthy to dead in about a year. So it
So
really,
this is a regime shift.
it's a complete, it's exactly, yeah, stable state to another one sort of game is what we would say.
Are there any data where you have systematic removal of the higher trophic levels so you can look for population fluctuations and other sort of early warning indicators of regime shift?
Yes, and I'll show you some of that. Yeah, not as good as you would really like in the dynamic world, but yes, we have a lot of examples over space as opposed to where someone's really followed it through as you overfish one. There are some, but not with microbial data. It's starting to be collected, but not a ton of it. I'll show you what we do have. And so what happens here, and this is just a general thing, and you'll also see this in lots of ecosystems where you change from macroorganisms to microorganisms, that you see that these microbes that start growing under these conditions look more like potential pathogens, is what we would call it. And by that, I mean that they're heterotrophs. They're super heterotrophs. They really like to eat stuff, right? That is, they like good food. They like hamburger, is the way to think about it. And I'll show you a little bit that there's this increase in coral interaction zones, which I think is interesting from a modeling point. And then a little bit of this kind of paradox where you're producing more of this, which actually makes the standing stock of DOC go down because you're producing so much microbial biomass. This is, again, is just an example of the 4 islands. I'm just going to use it as a quick introduction. Again, really, sharks, no small fish. Start to remove sharks, you get more small fish. You start losing them. Only small fish because we ate everything, and then the whole system collapses. And the bottom moves from coral and algae, to just macroalgae, with a very small amount of people. And the one thing that is time and time again is that your number of microbes, so this is the place without people, this is the place with people, the number of microbes go up about an order of magnitude roughly, and the number of viruses go up about an order of magnitude because they're just following, these are phages that are eating those bacteria.
Why can't I can see if you remove the sharks, the small fish increase bit.
Yeah.
Why can't they deal with the energy?
Right, because they just become big and fat and they don't eat that much more, and there's not that much more. They become habitat limited is the argument.
Right.
We're actively, or my collaborators are actively looking at that, but there is like this rule of big holes, big fish, small holes, small fish, and when the reef structure collapses, you lose that structure.
To what extent does eutrophication interfere? Because
Yeah,
and I talk mainly about the top-down effect.
Yeah. Eutrophication does just exactly the same thing, and we've got great examples out here where we can prove where eutrophication causes it instead of the removal of the fish. So we have places where boats have run into the reef, and the iron in this part of the ocean is so limiting that you get a dead reef around just where that one nutrient it releases them from a bottom-up control. Yeah, and it's really clear, like it's very solid. So that would for those of you you can imagine that if you push in enough nutrients, you can actually get over where you can feed these guys so much that they feed the microbes and then they kill off the coral. It's the same mechanism, it's just a different way of getting there. And usually on reefs we do both. That's the problem. Okay, so the microbial numbers go up by an order of magnitude, and they also switch. This is just metagenomic data again from an earlier study. And all I wanted to point out to you is that you go through this thing where you seem to see more autotrophs like cyanobacteria, a different type of cyanobacteria than what we see on a healthy reef becomes more apparent. And then there's this whole regime shift where you lose, where you gain all these microbes, and in particular you gain this fraction, which we would call opportunistic pathogens or potential pathogens. And these are real pathogens. These are the marine equivalents of E. coli. I mean literally, they're just bad guys. And the other thing that is kind of the nail on the coffin, so to speak, and this is a whole bunch of other, this is just a limited version of it, but this is a hard coral cover going along this axis. This is the prevalence of healthy corals going on this axis, right? Unhealthy corals on this axis. And what you see is that in places where Christmas, where things are dying, even though you have almost no corals left, they're almost all sick, right? And when you have a lot of corals, which in potential hosts, they're much healthier. So this is exactly what you expect with an opportunistic infection as opposed to a specific pathogen moving through the system. All right, so the ecology, like in the big model of it, says this, that on a healthy coral reef like Kingman, your primary production is building this bottom and feeding these guys. And on a screwed up one, your primary production is destroying the structure actually and feeding the microbes, is what it looks like. And so how general is it? Here's the Pacific, and these are all American protectorates. And so we have data sets from almost 100 sites and 29 islands out there. And I put this in here mostly for you, Chewy. So this is some energetic stuff. So what we do is we go down and we collect water from the bottom, and then we're going to take that up and we're going to count the bacteria. We're going to get their biomass right off the reef. And then the fish guys are going to go along and they have a transect tape, because this is what you do in marine biology, and you swim along and you count the number of fish in this amount of the water column. So we end up with equivalent numbers of biomass and kind of species-specific biomass in the end. And then the best tool that we still have, because it's really hard to do microbial growth rates, so when we do it, we for the most part have to use radiation and so forth, and that's not so great for large scale surveys, because you can't really use radiation on as many places as you'd like. We do some calorimetry, which Andy's been involved in, but what we're going to do in this case is just extrapolate. So what we use is this metabolic theory of ecology, which relates biomass to wattage, power required by an organism. And this is Brown's work, if you're familiar with this. And we just say how much energy is required by the microbes and how much is required by the fish in this particular piece of water on this coral reef. And then we come up with what is a microbialization score, which is the total metabolic requirements divided into that required by the microbes. This is to give you a rough feel about how it works, how it would look. So this is total metabolic rates for a 10 meters of water going up, so 10 meters high. And this is the watts per that unit volume. This is a microbialization score going on here. And it depends on where you are, you get more or less total wattage is produced. And that's sun-driven for the most part, right? And there'll be nutrients will run into that. And then you can see that there's quite a bit of difference in whether you have like few microbes and mostly fish, or to someplace like Hawaii where all you have left are microbes in the system. Okay? And the rough, by rule of thumb, is that 1 gram of microbes in this same amount of water is going to be equivalent to 500 grams of fish biomass. approximately. It doesn't take many microbes because they're spinning so quickly. And 500 grams of fish is really a lot of fish. That's like a really, really healthy reef would have something in that range. And I'm going to compare it against just a cumulative index of how much human stuff is going on. So this is how much, this is a metric that came out of the NC's group. And they just put all this stuff together and say, these are things that we think screw with the coral reef, and they come up with a number. And when you plot it, what you see is that this NC score versus the microbialization score are really nicely correlated. And in fact, the microbialization is doing a much better job, because you'll notice here that Johnston et al. Has a high microbialization score, even though it wasn't predicted to be in that bad of shape by these. This is where we dumped all of our, most of our atomic waste and burnt all of our bio warfare agents and so forth. And Agent Orange stored there. It kind of looks like an aircraft carrier because it's been so retrofitted from the sky. So it's like really actually pretty messed up. And then Wake Island, for some reason, ends up as with a bad NC score. It's actually one of the nicest reefs. So it's doing well with the microbialization. So we think you can get instant fills of the health of a system just by looking essentially at the energy requirements of the microbes compared to the fish. Now, so that's a Pacific-wide trend, and we're also seeing it in other parts of the ocean now. The other thing that's really exciting or interesting is that, remember, I told you that DOC production, even though you're producing a lot of more photosynthate in the system, and your higher support of the microbial biomass means that actually this standing stock goes down. And that'll be like maybe 5 to 10 micromolar in the whole ocean of the coral reef. So this is an example of where we're going to take all of the data. This is Andy's work, where over about a decade, we've been looking at microbial dynamics on coral reefs, and we're going to get the microbes and measure the amount of dissolved organic carbon. We've got metagenomes where we've been sequencing different things from all over the place. And then we also have the fish counts and how much is living on the bottom. And it's about 200 sites from all over the world. And the trends are ecology, so you're gonna have to deal. So this would, what he's plotted here is the amount of benthic cover and then the amount of dissolved organic carbon. And as this changes, as it moves towards algae, you actually are getting less DOC. So even though you're producing more DOC, and we know this for sure, you're actually getting less. And it's because we're getting more microbes effectively. Does that sound good to everybody? So there's a problem here, and this is something that the microbes eat into DOC, which we don't know it very well chemically, what's really going on, but a lot of dissolved organic carbon in the ocean is not very readily accessible to the microbes. So it's like refractory, we call it, and that refractory stuff is actually being chewed up in these messed up coral reefs.
Can I ask a question?
Yeah.
Do you have any idea to what extent the DOC might come from the terrestrial though?
No terrestrial inputs here.
Okay.
Yeah, well, sorry. In some cases we will. In this survey we would have terrestrial, that's right. In the Line Islands there's no terrestrial input. Here, yes, we would, because we would have the nitrogen isotope and carbon isotope stuff. We could tell you how much is coming. Yeah, but I don't know the answer. Yeah. The other thing that that Andy's been looking at is then what are they doing in these different systems, and this is, I think, really cool. I'm hoping it's cool. That glycolysis is what's driving metabolism, microbial metabolism on the healthy reefs, and they switch to the pentose phosphate pathway, PPP, when they're on this. And what that means for those of you that don't care about any of this stuff, it means that this is the pathway this is your efficient pathway, this is your inefficient pathway. And by that, the amount of fixed carbon you put into the system, you get less ATP out of it, let's say it that way.
But it's faster, right?
It's faster. Yeah, that's its game. Yeah, perfect. That seems to be it. It's just cranking away.
Right.
They don't, it's like, it is one of those things. Screw it, I'm just gonna eat as quickly and waste
Because
as much as I can.
It's
Yeah,
panicking.
yeah, yeah. So that seems to be what's going on there is PPP, they switch to it. And I think that that's your argument, right? That you're making in your paper. Good.
Right, and also the ET, which requires a lower activation energy to break down the carbon, so it's way less efficient, but as you said, you just eat faster and with less initial energy input, you get less out of it, but you don't care.
Right, right, exactly. And I think that what we're seeing in this case is that we've replaced the shark shunt with a microbial shunt, right? So now you're just pulling things because they're just chewing away, and that's reinforcing
So do,
an alternate stable state.
so
It's
do you
a-
understand how that switch, the metabolic switch works in detail? Or is it,
Is it
No.
a switch, or is it just a replacement? That would be consistent with the Pseudomonas and Vibrio's.
It's more of an ecology thing. Yeah, it isn't. Yeah, I mean they can. Different bugs can switch between the two, but it's if it smells like ecology, it's just community changes. Is my guess, right?
what's the difference? Because I'm not trying to do the difference.
Well, because you can force some bugs to like use one pathway or another under laboratory conditions, but what you actually see is that when they're in one energy regime, that one guys that can grow really fast just replace the others if there's a lot of energy, or they're not as efficient and they don't do as well when there's limited energy. Did I say that correctly?
Why is that different from a switch?
Sorry, it's a switch, but not with the same bugs. It's different bacteria.
It's different, yes.
Yeah.
But the ecosystem is
Yeah.
switching, that's what I mean.
Yeah,
The community is switching, and different players are coming into play.
correct.
So it is a switch at the ecosystem level.
Yes, at the ecosystem level, it's definitely a switch.
And we actually have a Pseudomonas that's a plant pathogen that's swept around the world recently. And on an island, it carries enolase, which is the penultimate gene in glycolysis. And the purpose of that seems to be to upregulate glycolysis to tip more into the TCA cycle, which allows, and the Pseudomonas subverts the plant and gets TCA intermediates out of the plant. So it's actually manipulating the host to allow it then to compete using a more wasteful strategy to outcompete other organisms. So it is possible that you could actually get, what you had in mind first, perhaps a metabolic shift.
Yeah.
The organisms themselves
Yeah.
driving
Right.
this change.
Yeah.
And that's really important because that all occurs in those carboxysome sort of looking structures, which is the quantum thing that I totally figured out that's going to revolutionize all of biology. Okay.
You're doing so well on something.
The lies. You can only tell so many. All right, so this is the last bit, and I'm just going to talk to, because I think from the point of view of math and modelers, this is one that's you can get a real handle on, and I think is one of the main dynamics, which is that, remember, you're getting an increase in interaction zones. So everything I've been telling you is really how the water is changing, right? And that's mostly, we think, just things are coming off the reef and we're measuring it. And that's what we're seeing. And the game is really these things sitting on the bottom competing with each other for space. That's what we think is, or that's how we envision it. And this is just what it looks like. So a reef is incredibly complicated. These guys, this holobiont is competing with that, but I'm not sure they're actually different holobionts, but whatever. These guys you would love. Do you see this pattern? That's one of those hexagons they form because of the, so these are the same species, and they actually put themselves into these perfect patterns that you can see. So these guys are all competing. So this is where the algae and the corals and the sponges, et cetera, would all be looking for space on the bottom. And in the simplest terms, what that means is that the bigger you are, the more area you have fighting a smaller circumference effectively. effectively. So you can put more energy into fighting at the edges. And so this gives you a very easy geometric-based positive feedback for coral replacement by algae or vice versa. Okay. And I hope Christian and Andy are going to work on this a little bit. This gives you a little bit of the data. We were jealous of the fish guys because they got to use tapes, so we got tapes too, and we would put them down on the benthos, and then we would go along, and every time you find a coral, which this takes a long time in some places, you say, okay, what is that coral interacting with, and is it winning or losing, right? And so that you can imagine this takes several hours at a time, and you just record the percent and so forth, and you come up with a metric of how often is a coral losing or winning. And this is just what it looks like, right? So here you can see that this is the algae, the coral is winning, right? And in some cases they grow right next to each other, so we call that a neutral interaction. So they seem to be, they've decided not to fight right there for the moment. And then other places, then you can see the algae going over the top of the coral, right? So you can just go out there and count the stuff. And when you do it, what you find is something like this. So this is curacao, for example, and as the coral cover changes, you get different interactions with the CCA, same with turf algae, etc. But what's important is the win-loss ratio. So in places like the Line Islands, if you don't have people, which are these islands here, you don't, the coral will actually win against the turf algae. But if you have people where we've overfished it, these islands here, the turf algae win against the coral, right? And it's really clear. There's not much doubt. And this is what's going on at these interaction zones. So here's an EM. So this is your coral animal here. This is the interaction zone, and the algae would be here. And what's going on right up against the side of the coral are these basically microbial mats piled up right against them, and they're filled with heterotrophic and autotrophic unicellular organisms, right? Everything from, and they just look like piles of stuff. And it reminds you a lot of it. I'm not going to call it a biofilm, but you know, the things you get them in wounds, you get them all over the place, and you can just imagine basically what's going on is that these guys down that are deeper in there have turned the whole thing hypoxic, right? So they're just eating up all the oxygen and they go to fermentative. And this is just a simple experiment. This is marine biology now, guys. So we take and we put things together. It's always kind of crude looking. So what we've got is we've got an algae across from a coral, and we put a disc between it. And the disc only lets dissolve things through. So viruses and bacteria can't make it through it. And when we do this, if you have the coral by itself, it's happy. If you have the coral across from the algae, all of the corals die. And then if you do the same experiment, coral by itself is happy. Coral across from algae all live if you add an antibiotic, right? So something is dissolving, which is helping feed the corals, or feed the bacteria, which kill the corals. It's the hypothesis. And the other thing it's associated with is hypoxia. So as the coral dies, it just becomes hypoxic in these zones that are across from the algae. This is the same thing. This is just a natural experiment. So what you see here, there's the algae, here's the coral. This is what one of these interfaces would look like. And if you go with a microprobe in this case, and you just look at different places, when you're on the algae, it's superoxic because it's doing photosynthesis. When you're on the coral, it's also superoxic because it has the algae doing photosynthesis. And then in the interaction zones, you actually get the hypoxic zones. You get these hypoxia zones. The one exception is, remember I told you, there are these guys here, these CCAs. And CCAs do fight with coral a little bit, but they actually seem to be pretty benign. So this is the coral, the interface, and the algae. You get increased oxygen, like I said, because they're both photosynthesizers. And you get hypoxia sort of readings at the interfaces, except for when you're interacting with the CCA. So that thing that's part of the normal healthy reef seems to be, they don't fight in the same way, at least. And it's dependent on the algae, because if you just go scrape it away, you get, you lose the hypoxia. And the same thing, if you just treat it with antibiotics, you also lose the hypoxia. All of this points to just a microbial thing. And I'm going over time, so I'm going to stop, or I'm just going to tell you quickly. Also, if you look at these interfaces, they have different microbes. And this is a measure, which is a kind of a nice one. The corals, remember, corals are cnidarian, so they naturally have GFP in them, so green fluorescence protein. And then this is the algae. So you can see them fluoresce. And GFP is actually dependent on oxygen, right? So you can just look, and you can just go in the field and say, do they feel stressed in terms of their oxygen dynamics? And you see that when they're next to turf, they're oxygen stressed. When they're not next to turf, when they're next to CCA, they're not. And this is wow, I'm just beating you to death with this. So this is what it looks like under an octode. So you can see that you get these hypoxic zones. So guys, it's not showing very well. The reds, the darker the red, the more hypoxia. So these interfaces you can see in a whole bunch of different ways.
So are you saying that there's a higher abundance of microbes in these interfaces, or that there's higher diversity?
There's both. There's actually higher diversity and there's higher activity by a fair amount.
Right.
And that...
Is that surprising?
No, it isn't. Not from a microbial ecology point of view. It's very surprising from the coral reef field. They have a very hard time dealing with microbes. And so they never made a connection between why removing the fish grazing would actually kill a coral. And it's basically microbial driven. And one of the things that we're trying to do with the modeling here is to actually get this down, because they don't understand things like boundary layers, right? So they don't believe that there are places, because there's a whole bunch of water running by, so there's no way it could be. It's the same thing in like the cystic fibrosis work. We argue whether you can have hypoxia in the lung, right? Because lungs can't be hypoxic because they've got air in them. Yeah, so it's an exact same thing. And this is the question really that we're left with, is that how do you maintain these hypoxia environments with high flow? It's going to be boundary conditions are going to be the cause, but we need to prove it, actually just get it down and say, this is how fast the water is moving, this is how much hypoxia we see, this is how many microbes it's supporting, that sort of thing. Okay? And then maybe if we could work in the switch of the efficiency that they're using it, that would be wonderful.
By the way, do you have any idea how thick the boundary layer is?
We do, but I don't remember. He just asked us that. So, yeah, there were a lot of microprobe assays done by people, and they've done them under high flow and low flow conditions and so forth. So we do have a reasonable idea about what the boundary conditions look like. There's also centimeter resolution where they use one of the sonar systems. I'm not sure which one, what's it called, but you point it down at the bottom, and then you can watch different packets of water move, and you can get about centimeter resolution. There's that also for coral reefs. All right, and then I will just thank everybody. This is, of course, a giant group of people that have done this. Andy is here, so you can talk to him about a lot of it. This is the lady that actually runs my lab and is the brains behind everything, and she did all of the metagenome work that I presented at the beginning. This is my lab, and these are the math guys who do all of the really, they keep us out of trouble, actually. Very good group. Peter Solomon and Jim Knowlton had some of their work in here. Rob Edwards is my longtime collaborator for working on bioinformatics. Anka is here, and she's worked a lot on some of this data. And then these are my collaborators at mostly at Scripps Institution of Oceanography, Stuart Sandin and Rick Solid, Jen Smith and BZ. And they're the ones that actually get us out to the middle of the ocean and get us back. And this is what I mean by there's a lot of sharks up there. And then these people pay for it. And with that, I'll stop so we can not cut into someone else's time. Why don't we switch and you guys can ask questions? Don't we have someone else speaking today? Oh, all right. But then we're cutting into beard. All right, so let's go ahead. Do you guys have any more questions? Yes.
So is this a one-way switch, or if you stop fishing, do the fish reinvade? Does the coral recover? And how
Yeah.
does that process?
So far, no. We've never seen it do that. So it does seem to be one of those ecological things where you're going to probably have a whole bunch of successional stages to go back to the original climax community. No examples of it so far. We do know of places where they've removed like a really like a point stress, and in that case you usually get like one or two coral types to come back, and then they dominate. In the one example we know, it's basically just dominated by one particular coral type, and it's never really bounced back to anything approaching the climax community. Right? So it's going to be, from the engineering point of view, so conservation, I think now we know exactly what to do. Restoration, I'm not sure that we have it. Well, I know for sure that we have no idea what we're going to do and how we're going to get that to work. Yes.
I'm trying to see whether I understand the full picture of these small scale interaction between the corals and the algae, and especially the oxygen depletion.
Yeah.
So what is the depletion of oxygen? What is happening there? Is it that they eat the dead tissue of the coral, or do they eat the
So it's a
organic
little
matter of the
chicken and the
algae?
egg. They are eating the food that's being produced by the algae, and then as they eat the tissue, then they get ramped up even more. It's
So
our
they feed
model.
on both the tissue of the algae and the dead tissue.
Exactly.
Of the
Yeah.
And are you really sure about this, or is this hypothesis? Or?
We know that we can. No, we're not very sure about it actually. We know that all of that is happening, but we don't really know
Like
the gut.
quantitatively?
Yeah.
Okay.
Well, we know quantitatively what's going on too, but we don't know that it is chicken and the egg game, right? Because we don't know, is it just one bacteria, particular type was cultured up by the algae, which then led to the system falling apart, which is totally possible, right? That happens in lots of diseases. You get, there is something that causes it, but you never really know that was the cause. You just get into another system that's running.
But it would be a key question, right? Because if it's a specific organism doing this, or whether
Right.
it's just a bunch of organisms, it's a totally different way.
Well, it's the mat crawling across the reef is a bunch of organisms doing it. The initiating event has never been caught. I mean, we've built ones, and we know that certain stressors favor Vibrios, for example, or certain Pseudomonas, but we don't know of anything that's like a pathogen that starts the game, right? And that's where we are. Anybody else?